Okay, so today we're covering Lab 21. It's a pretty long lecture, so we break it up into 3 parts. Right? And so what you need to keep in mind when we're doing this is don't get overwhelmed or don't feel overwhelmed. Just keep in mind the structure, location, and function. Basically, where is it? What is it? How does it work? Try to keep it under. You should be able to give 1 sentence, 2 sentence definitions for all these things in terms of function. Again, just don't get too overwhelmed by anything. All right, and so like I said, we're going to cover 3 parts today. Part 1 is going to cover some basic terminology, some of the gross anatomy structures, and then 2 specific structures within it, the meninges and the ventricles. Part 2 is going to get into some of the lobes and other internal structures, and then Part 3 deals with the cranial nerves. So just like in Chapter 2 when you had to learn anterior, posterior, superior, inferior, superficial, deep, there are specific directional terms that are associated with the brain. Right? So rostral means anterior, towards the front. Caudal is like a tail. It means towards the back or posterior. So rostral is anterior, caudal is posterior. The main meat of the brain is called the cerebrum. Right? This composes almost 85% of the actual brain volume. The other 2 parts are the brainstem and the cerebellum. The brainstem is shown in green, the cerebellum in blue. Cerebellum really it's a very small portion of the brain, but it contains up to about 50% of the neurons, and that's because a lot of motor coordination occurs in this area, occurs within this cerebellum. Some other main structures that we want you to know: when you look at the overall kind of structure of a brain, you are looking at mostly the cerebral cortex. It's the top 3 millimeters of the brain, and this is composed of mostly of gray matter. And so gray matter, the way to think about that is that it's the computer. So this is the action sites of the brain. This is where most of the activity occurs. And then you have white matter, which are basically myelinated axons that act as cables between the computers and other structures to transmit information. And so again, the myelination, you really don't need to be too concerned with it. It's just kind of imagine it as insulation for cables. It's really keeping the electrical signals that are passed along kind of running smoothly and pretty focused along those pathways. So again, the cerebral cortex is made up mostly of gray matter, or of the computers. And within that cortex, you have what are called convolutions. And the convolutions are those folds within the brain. And so again, whenever you see folds, basically you're increasing the surface area, right? So it's the same idea as a microvilli. If you guys remember way back to Lab 6 when we were talking about, do you guys remember the epithelial tissue, right? That simple columnar has that epithelial, or has that microvilli on it. All right, so same sort of context here with the cerebral cortex. All these folds are increasing surface area, and the convolutions, or these folds, are composed of both ridges and depressions. And the ridges are a special name. Gyri are the plural, or gyrus is singular, and then sulci or sulcus are the depressions. Sometimes you see them called fissures, but they're used pretty interchangeably. Right, so the convolutions, which are made up of gyri and sulci, which then form the cerebral cortex. And if you look at the brain, this kind of superior view, looking down on it, is you see 2 halves, or 2 hemispheres. You have a left hemisphere and a right hemisphere, and along the middle you see a very long line, depression or fissure, and this is the longitudinal fissure, which effectively separates the 2 hemispheres. Alright, so we've talked about some of these general structures around the brain, and now we're going to get into some of the specific structures that help support it. And the first one we're going to talk about is the Meninges, which is composed up of 3 layers: the Dura matter, the arachnoid matter, and the pia matter. And so the Dura matter, which is basically dense connective tissue, is actually composed of 2 individual layers as well. Further, I guess, it's the periosteal layer and the meningeal layer. The periosteal is going to be superficial to the meningeal. And so you actually can see the Dura matter surrounding the brainstem when you do the sheep dissection, the sheep brain dissection, and it kind of looks as this really kind of plasticky, opaque look to it and feel to it. It's tough. It's really kind of this outmost protective layer. If you go 1 layer deeper, you get into the arachnoid matter. In itself is a thin membrane, but the subarachnoid space that it creates between the arachnoid matter and the pia matter is where the CSF is. And it has the arachnoid, as you might guess from that word, it's very spidery or web-like in its structure. All right, so that subarachnoid space in particular has this web-like structure among it that allows the CSF to pass through. And so CSF is cerebrospinal fluid, and we'll talk about that in the next couple slides. The deepest layer is the pia matter, and this is going to be the one that's going to directly cover the brain, basically acting like saran wrap, or in the sense that it is shrink wrapped around the brain very tightly. It's another one you can actually see on the sheep brain. You kind of pick at it. When you're making these cuts, you actually you can feel this thin film over it, and that's the pia matter. And if you remember that the Meninges we kind of the last time we saw that term pop up is when we were talking about the distinguishing between cranial and facial bones, right? So cranial bones are going to be the ones that are in direct contact with the Meninges, and the facial bones are going to be the ones that are not. Here's a coronal cut of the cranial meninges showing kind of a section where we're looking probably anterior. And you can see the Dura matter with the periosteal layer and the meningeal layer that it's composed of. And you can go down a little bit deeper and see the arachnoid matter and the arachnoid villi that kind of compose that subarachnoid space where the CSF fluid is passed through. And then you can see that in the purple, the pia matter. So meningitis is going to be, it can be bacterial, it can be fungal, it can be a viral infection of the meninges. And so it basically typically enters the brain or enters the area through the choroid plexus, which is a structure that CSF is created and secreted into the brain. And it's interesting when you think about an infection of the meninges, or what an infection is, or what kind of the symptoms that are associated with it. But basically, when you have an infection, it's going to lead to inflammation. So your body's trying to fight this infection, so it's going to pump fluids into that whatever specific area. In this case, it's the surrounding of the brain, and leading to inflammation. However, since this area is kind of this packed shell, right, you can't really expand. It's not going to swell. The only place for it to swell is in on itself. So you lead to this inflammation of the brain, right? So the swelling of the brain. And the blood-brain barrier, which is this kind of filter that allows things in and out, can't really function properly. And so it becomes overwhelmed, and so you have this chronic inflammation and basically all this pressure on your brain cells, and it leads to death or apoptosis, in the sense that these cells are not used to this pressure, and that leads to cell death, and without treatment, basically death, effectively. And so it's kind of a big deal, and you see it pop up in areas where people kind of are living together for the first time in high densities. So college campuses is a really kind of characteristic example of that. We had a scare at NAU last year and a couple of years ago, and so it's kind of a big deal in terms of treatment, mainly because it's fatal, the severity of it. All right, so we've talked about the meninges. The other main structures that we're going to talk about are the ventricles, and these are associated because they're producing the CSF, or that cerebrospinal fluid, that basically travels throughout the entire central nervous system and passing through that arachnoid villi until it gets reabsorbed, right? So it's actually existing within the meninges. All right, here's a diagram showing an anterior and a lateral view of the ventricles. And so this I kind of think looks like the Starship Enterprise or some sort of spaceship. The ventricles make up some sort of spaceship that exists within the brain. And you have what I call the left lateral ventricle, the right lateral ventricle, the third ventricle, and the fourth ventricle. And again, these are all dealing with the production of CSF. And you can see between the third ventricle and the fourth ventricle, that's what's called the cerebral aqueduct, right? And if you remember what an aqueduct is, it's just a little passageway that allows fluid to go through. And usually you see it in terms of water in cities back in the Roman times, but here it's obviously referencing the connection between the ventricles or the flow of CSF. And CSF itself is basically this clear fluid that the brain produces, basically a ½ liter every day, and it's getting produced and absorbed, and it's really dealing with maintaining a neutrally buoyant and chemically stable environment, right? So in all sense of the word, it's kind of offering protection for the brain. So kind of making sure that waste products are moved away, and that brain doesn't slosh and hit against the side of your skull. And so the way the specific structure within the ventricle that's producing this is called the choroid plexus, or in Greek or Latin, the delicate knot. And so that's the specific site of CSF production, and you'll be able to see this when we do the dissections. And a way to think about the functions of CSF is with a hangover, right? So you've had one or you've heard of one. A hangover comes when you drink too much alcohol, and the next morning you wake up with this pounding headache. And one of the reasons that's causing that headache is this lack or this difficulty in producing CSF. So alcohol itself is a diuretic and makes you go pee a lot. So you become dehydrated. And when you become dehydrated, your body really struggles to produce CSF. And you see that turnover of 500 mLs a day of CSF, and your body's really, if you don't have enough fluids there, it's really struggling to produce that CSF. And when you're not able to produce it, you're basically, those waste products are sitting on your brain longer. Every time you turn over in your sleep, your brain is kind of hitting against the side of your skull, and so you kind of don't have the balance. So you need to make sure that if you choose to drink, that you hydrate properly. So you drink water constantly. You're drinking Gatorade or whatever to help maintain the electrolytes and all the kind of important properties that go into CSF, the production of CSF. Here's a schematic showing the general flow. If you look at, again, this is a lateral view. If you can see through 1, 2, and 3, you have basically the first 3 ventricles, the left and right lateral ventricle. In this case, it would be the right ventricle. 3 is the third ventricle, and you can kind of see those wavy structures are all the choroid plexus. It passed through 4, which is kind of the cerebral aqueduct, to the fourth ventricle, which is located just deep to the cerebellum. And then when you look to 7 or 8, you're dealing with a distribution and then reabsorption through the subarachnoid space, right? So that arachnoid villa. A disease that you see with the ventricles is hydrocephaly. And that person's head is swollen because if you remember that the cranial bones haven't quite fused yet when you are born or when you're young, and so your brain's still developing. So what's happening here is that you have an overproduction or a blockage of drainage for the CSF fluid. And so if you're dealing with CSF fluid, basically what's happening is it's very similar to meningitis in the sense that you're putting pressure on the skull, and the blood-brain barrier and all these things can't really deal with it. So you put pressure in the brain, leads to cell death. But you don't really see that too often in first world countries anymore because we have the medical technology to deal with it, and we can watch some videos in class of a shunt, or you can Google it yourself, but basically a shunt being put in that will relieve the pressure, and they will run a tube that drains into the stomach and then is passed naturally. Right? So there's ways to deal with it now, deal with either the overproduction or the blockage of CSF. And that's it for part 1.